Hello everyone, this is Anita. I'm a faculty member at University of South Carolina, upstate, and the work I'll be presenting today is a collaborative work between myself and Dr. Homeo Falafer's lab at USC Columbia and his student, undergraduate student Niharika Pandala. Before I begin, I'd like to thank ASBMB for selecting our abstract and also giving us the opportunity to present through this virtual platform. Now, ironically, we study a coronavirus. So we have been studying non-structural protein 1 of SARS coronavirus for the past 3 years. And the reason we were interested in this protein is because this protein shows a lot of similarity with proteins from other viruses, but it has also something very unique. So severe acute respiratory syndrome coronavirus, or SARS-CoV outbreak, started in China in 2002. And during this time, it actually spread to 27 different countries, resulted in 9.6% fatality rates. Now, this is a RNA protein with a positively stranded RNA genome, really large, about 30 kilobase long. And one of the non-structural protein is non-structural protein 1. The uniqueness of this protein is not only this is structurally very flexible, that makes it challenging to study this protein. And second, if you just express NSP1 by itself, it inhibits host gene expression. And then Jauregui Wei lab has done mutations on the surface residues of this protein to show that they significantly alter its function. There are some mutations that attenuate NSP1, and then there are other mutations that aggravate its effect. So together, what the two things we wanted to study, one is a proteomic analysis of NSP1 and its mutants to find what are the host proteins that are functionally relevant to aid NSP1 in its host shut-off activity. And second, computational modeling to identify if there is any structural topological change on the NSP1 surface that will allow us to analyze why different mutations behave differently. So let me give you an overview of how this protein acts. NSP1 binds to the 40S ribosome and halts it from assembling any further to the complete ribosome. So protein synthesis on host mRNAs are stalled. Not only that, in a 1-2 punch, this protein also binds an unknown endonuclease and results in cleavage of the host messenger RNA. Results in cleavage of host messenger RNA. Now, surprisingly, NSP1 still binds to the viral RNA, but because of a hairpin loop structure in the viral RNA is not cleaved by the same mechanism that allows NSP1 to cleave host messenger RNA. Now, there are different mutations that are studied so far. One is arginine lysine mutation that allows NSP1 to still stall ribosome assembly, but it cannot bind to the endonuclease, so cannot cleave the RNA. The other mutation, lysine histidine mutation, does not stall the ribosome or cleave the RNA, so it can be used as a complete inactive mutant. So using these three proteins, the wild-type NSP1 and the two mutants, we wanted to identify host proteins that bind to NSP1 and its mutant. So we did bacterial expression of the protein and then incubated it with whole cell extract and pull down these complexes and send out specific excise bands for identification of proteins by mass spec. And I'm not going into details of the whole mass spec analysis. I'm going to show you one specific example that was identified by the mass spec. We were surprised to find nucleoporin NUP93 associated with NSP1, but the association is weakened by the lysine histidine mutation. So NUP93 is part of the nuclear pore complex, which sits on the nuclear envelope, acting as a gatekeeper between the cytoplasm and the nucleus, allowing only specific cargo to come in and out of the nucleus and cytoplasm. So we first asked what happens to NUP93 in the presence of NSP1. So Garrett Gomez, undergraduate student in my lab, did immunofluorescence to look at the localization of NUP93 in the presence and absence of NSP1. So in regular cells, when we do not express any NSP1, NUP93 is present around the nucleus, showing this nice and clear leveling and the ring structure around nucleus, confirming the presence of NUP93 on the nuclear envelope. When we express NSP1 for 24 hours, we do not see this nice and clear ring pattern anymore. Most of the NUP93 signal is found inside the nucleus. And then when we express the inactive mutant that doesn't bind to NUP93, we again start seeing this clear ring structure, confirming that NUP93 is now present on the nuclear envelope. We took the Z-stack and quantified NUP93 fluorescence intensity under these three conditions, and again confirmed that NUP93 is present inside the nucleus in the presence of NSP1. So we can conclude that NSP1 binds to NUP93 and alters its localization from the nuclear pore into the nucleoplasm. So we also wanted to know that how this one mutation that changed how NUP93 binds to the nucleus, to the NUP93, it relates to all the other mutations known for NSP1. So here is a list of mutations that were published by Jauregui. And the first set of mutations shows no difference in gene expression. So remember again, NSP1 binds to host messenger RNA, stalls protein synthesis, cleaves the messenger RNA, leaving no more template for further protein synthesis. So in this experiment, they were measuring how host shutoff activity by NSP1 was affected by these mutations. So the first set of experiments shows that gene expression was unchanged by these mutations. The next two columns shows how NSP1's activity is suppressed by this whole set of mutations. So this was one of the mutations that I showed you earlier, how this actually doesn't allow NUP93 to act the way it does with wild type NSP1. Now, the last column shows mutations that actually aggravates NSP1's effect on gene expression. So with these mutations, NSP1 is more active than the wild type NSP1. We were specifically interested in certain mutations, for example, this lysine 58 mutation that inhibits NSP1's effect on gene expression. However, additional mutation around this area actually aggravates this effect. So we wanted to study how this area can be how this amino acid sequence on NSP1 can be helping NSP1 to bind to host proteins or changes its structure to allow it to get weakened or aggravated upon mutation. So we wanted to identify if these mutations actually creates any structural or topological changes on NSP1. So I collaborated with Dr. Home Falafar at USC Columbia, and they started with iterative threading assembly refinement of NSP1. So this iTesser is a software that uses hierarchical approach to predict protein structure. By using available protein structure from protein database and threading the sequence of the protein multiple times to generate full-scale atomic model. It also gives you some confidence score. And now when you use the full length NSP1, we found that our confidence score was really, really low, pardoning to the flexible region of NSP1. So you can see that the C-terminus part of the NSP1 takes up different structure, and that's why it's generating multiple models with very low confidence score. So next, what we did is we actually used a truncated version of NSP1, which is the first 120 amino acid long, and this is the part which is more structurally defined. So using that 120 amino acids of NSP1, this is the structure that was created by iTesser compared to the full-length protein. And as you can see, this confidence score improves significantly. So next, what Dr. Volafer's undergraduate student Niharika did is using this model, she mapped different mutations on NSP1 surface and calculated if that gives rise to any change in steric or topological change on NSP1 surface. So I'll show you only one example, the K58E mutation. This mutation by itself actually decreases how long the amino acid side chain is protruded from the backbone. So what she did is, this is the protein backbone, and she calculated the length of the amino acids, the most extended part of the side chain, and calculated this distance, and she found that with K58 to E mutation, it actually decreases the side chain length by almost 2 angstrom. However, when the other mutations made in the same area, so E55R, E57R, or G59R, the lengths are significantly increased. We are continuing to do these calculations and then allow change in pH solvation to incorporate whether there are any significant structural change upon these mutations. So finally, we are just continuing with visual molecular dynamics simulation and molecular dynamic simulations to analyze these mutants, their binding energies under different pH and solvation. And then biochemically, we are tagging NSP1 interacting proteins to identify what proteins are interacting with NSP1 and corresponding mutants. Finally, I would like to thank my undergraduate students who worked with me so far, Dr. Falafar student Niharika, and my collaborator, Dr. Hume Falafar from USC, Columbia, Dr. Lauren Ball from Medical University of South Carolina, Dr. Kurt Gustin and Shinji Makino for providing me plasmids, and then SC in brief for support. And again, thank you all for listening to my presentation. Thank you.